harvesting this large protein and scaling it up to get enough of the drug to treat patients. And so this is something that Prothelia has tasked with to try and produce it. And if you want to ask Rich where they are with that, you can. I know that they have a platform cell line now that's producing the protein. And the hope is that they have a company that they'll work with to scale it up so that we can get towards that period. But my role has been trying to produce all of the preclinical animal studies to actually then set up the stage for a future clinical trial. The other drugs that we're working on are actually a small molecule. We did a drug screen with NCATs at NIH here. This is the National Center for the Advancement of Translational Sciences. We did a drug screen looking for a integrin-enhancing drug, and we identified several, including issue 9516, which I saw up on the slide yesterday. We only just published it this month, that data, in Molecular Therapy. And it looks like in a very preliminary studies in the DYW mouse model that we're seeing great efficacy in an FDA-approved variant of that compound. So there is already an FDA-approved compound that's for another type of indication. So we think we can fast-track that drug like a megapel maybe, into a clinical trial because we know a lot of the safety features of that drug have been identified. With that, we just have to complete the studies, the preclinical studies to set the stage and make sure that we're convinced that we can move it next. And a lot of these studies, we'd like to have them also repeated in another lab, just to make sure everything is working, and that's kind of critical. And then the other big issue that we face, obviously, is the costs of the animals. They're incredibly expensive. So the DYW mouse, we say about it and you think, oh, I can generate mice breed like mice, you get tons of them. But in fact, we have to breed them as heterozygote animals, and we only get 1 or 2 mice that actually have the phenotype, the disease, the correct genotype per litter. And even though mice can have a litter of 5 or 6 pups or up to 10, you only maybe get 1. And so you have to have a huge colony. And for each cage and that we have, we get charged $1 per cage per day, right? So my mouse bill, I can't say, can be between up to $40,000 per year. So I have to be able to bring that amount of money in to not only support all my students and all my staff, but also to support the animal costs. So we're just going to give you a picture of the things that we face at the research side of things as well.
Do you have a question for Dean? Does it work? It doesn't work.
No, I think they said just talk into it and it's supposed to be recording.
I see. No question for Dean. All right, so my name is Dwi. I'm originally from Indonesia, so I also have a bit of accent. And then I did my PhD in the Netherlands, so that's for 10 years. I mean, not PhD for 10 years, but I stayed in the Netherlands for 10 years. So that's another kind of accent. And now I'm in Canada, so that's add up to that. So I win.
You
win. So I'm from Dr. Ronnie Cohen's lab in the Sick Kids Hospital in Toronto. And we are working on a technology called CRISPR-Cas9, which is basically a way to edit mutation in the gene. So in the lab, we are working on Duchenne and LAMA2, so editing dystrophin gene and LAMA2. And what we have done, we tested in one of the mouse models for LAMA2-CMD called the Dwi2J. And we've shown that correcting the mutation can prevent the progression of the disease. So this approach is a little bit different because it is mutation specific. So what we will have to do moving forward is we need to assess your mutations that are hopefully in the CMDIR. So this is also a part that I need to pitch in on behalf of the Cure CMD. So we need the genetic mutations to be deposited so that we can assess how to move forward. What else do I have to say? And anything that Dean said with the mouse. I can only echo that, yes.
Has CRISPR improved the ability to create the mouse models that you need? Like I heard that was one of the benefits of CRISPR because you can create mutations how you need. Does it help with the efficiency of creating the mice that you can then use testing on?
Yeah, so the work in Duchenne, we use CRISPR to also create mouse model. For the LAMA2, we used the Dwi mouse that has a subtype of mutation called splice site mutation. So what the Dwi mouse has is an exon 2 skip. You probably are familiar with exon skipping. So in this case, exon skipping is the cause of the disease. So what we are trying to do, we try to put back an exon that was skipped in the mouse. We call it exon inclusion. So the story is about to be out in a week, I hope. Yeah, in about a week. So you will hear more probably from the Cure CMD as well. If I if I have my slide, it's gonna be easier for sure. But we can talk after this if I have my laptop here. If you're interested, yeah.
Well, I think the great thing about CRISPR-Cas9 is that it could be used to personalize a mouse for any condition. So if your child has a particular genetic condition, it's possible to create a mouse that exactly mimics that genetic condition, and then you could do testing. So and it's much more rapid than the traditional ways of making a mouse. So that is a way if you could have a personalized mouse model for your child, and then you could treat it, and then you could see more accurately, rather than we have fairly generic models that we use. These are ones that were generated which lack lamina alpha 2, but then may not exactly replicate what your children's disease are.
There's still some targeting options with targeting problems with CRISPR-Cas9.
Yeah, and that came up recently when they looked at off-target effects, and it looks like it not only hits where you want pretty efficiently, but it can hit other sites, and that is the big worry. I mean, CRISPR is fairly early technology, and so there's got to be a lot of tweaking of it, I think, in the next few years. So it requires some more experimentation.
Yep. Thank you.
Okay.
So I'm Sweta Girgenrath, and the trend is set already. I have accent. So I have an Indian accent, and then my children say I have a very warped Boston accent. That sounds bad, Mom. What do you say? But anyways, that's what I have. So my work, I mean, I've been working on LAMA2 deficient disease for almost 15, 16 years. Before you know, it's a long time. My interest has been always trying to understand the disease and different pathologies which are associated with this primary loss of the gene. And I also feel very fortunate. The first trial, anti-apoptosis, I think I was one of the ones who had studied this in this disease. So hopefully there's a path forward. So my thoughts are, because I have been looking at the different pathomechanisms beyond the primary defect, is trying to find different interventions, what would make sense, and what are the time, in the natural history that, let's just giving an example, if it's inflammation, when is the best time to, target inflammation as opposed to, let's say, the growth regenerative defects of the muscle, and with the hope that we will be able to identify the right window to be treating the right pathology. And even if we understand that there is a path for most efficacious treatment to be combinatorial, I know there's a difficulty to bring such a treatment to the clinic. So it will be very useful if we could identify those windows and trial them, at least in the mouse, that if we can, target the right window, whether we can do one drug at one time and continue with some, basic maintenance over the other period. So I'm doing, I've spent a lot of time trying to identify, like, systematically trying one drug at a time. And also my other hope is to, trying to repurpose some of the drugs and whether there are benefits while we are waiting for this breakthrough, if there are some other drugs which will just stabilize the disease over time. Oftentimes when we used to write our grants, so I'm saying several years, right? Oh, gene therapy is, far-fetched, or protein, therapy is far-fetched. But I think we are also at a time where these things will be real very soon. And there are like fabulous work in the preclinical space which is happening, and I think there is a path forward because all different pharmas, small and big, are getting into that, and they're serious about this. So I'm just trying to work overall towards the goal, basically trying to find a therapy. That's it.
Can you talk about a little bit about the potential of bringing possibly gene therapy?
Yes, I mean, that's what I just suggested. There are discussions, as you know, that there are smaller biotechs which are already going and having pretty successful, they have pretty successful preclinical data to move into clinic. For example, there's discussions going on in myotubular myopathy. So we also, in MDC1A or LAMA2, there are very strong preclinical trials with not only transgenic, delivery of the gene, but also people have used AAVs to deliver this, and multiple labs have shown the outcomes are pretty convincing and phenomenal. So, and there is some discussion right now to see if, bigger and smaller pharmas would be interested to go, because of such a strong preclinical data, very strong animal model. These are all working towards drug companies to be more motivated to go towards that direction. I mean, I don't know whether there is any commitment anywhere yet, but people are thinking about it, thinking strongly.
Actually, that's a good point. I wanted to bring up the sort of relationship that everyone has. So, we're academic researchers. We do the basic research in our academic labs, and then what happens next? This is it's a really big group effort. So once you have identified something, then you often track to small pharma, right? Because they're ready to take the risk. And then if it looks good there, then you start to attract big pharma, right? So for example, the Pfizers of the world and other people to start bringing in their muscle to be able to move it forward. But on top of that, you also need the physicians to be on board as well, and you need the patient population as well. So, all of these elements have to come together to move it forward. And although a lot of us at the very beginning and entering into those phases, the path forward for us is there. We're sort of trying to track it forward, so we know which way we want to go. But it is definitely a big effort to bring all these different entities together. Everything has to work together at once to make it all work to bring the drug to trial.
On that note, I would also say. So I jumped quickly into what my research is. So, but my personal path was I was an academic researcher for many years, but due to some change of direction, I went to a big pharma, Pfizer. But my only reason, one thing which was I would be very, I think fortunate, when I was talking to somebody in big pharma, my only request to them was that, look, this is a very passionate thing for me, to my disease area, and I would like to continue working on it, and they supported it at some level. Yes.
Yeah.
I would say, like last year when I was starting, I was told very modestly that, okay, right now we are not interested, we are more interested in Duchenne space. But we have had CMD Biology Day focused LAMA2 in May at Pfizer's cost. They paid for everything. So I think they are already listening, and I would imagine that going forward we have rare disease days. I would really invite patients to come and join us, and I think when people put a face, and then things become a little different. Right now they just don't discard LAMA2 because they know I'm sitting in the room and they know that I'm so passionate about it. So they always say, well, we know Sweta is very passionate about it.
And persistent.
Persistent and passionate, for whatever the name is. But at least it's on the table. We are discussing this. On 21st of this month, I've been given 5 minutes time to pitch for my disease, and I will. So that's all I can say.
Is there a chance that a disease or an illness with a much larger population, such as cancer, can make a path forward for, say, gene therapy or CRISPR, and that will make using that as a technology to treat LAMA2 more economical or quicker to get through FDA clinical trial?
So I think Pfizer, we are at a good spot right now because they have gone into gene therapy with Duchenne at this time, but they have, because they are in this, so we are in the rare disease group. So it has been agreed upon by higher leaderships that rare diseases is where they want to see their gene therapies to proceed. So they are looking at other indications. So I mean, they won't go into cancer for gene therapy. They would rather look at the genetic diseases, and so autosomal recessive disease is on their horizon. They are thinking about them. So I think from that point of view, I think it's a very opportune moment to be around in that area and bring our disease in the forefront. Again, it's understood not even if they have absolutely 100% good intentions to be everywhere, we have to realize they may not be able to be everywhere. So that is understandable, but definitely being recessive, being autosomal rare disease, we are being on the table right now in the discussion.
Can I add one more on that? So the strategy that I'm developing, or our lab is developing right now, it's actually taking from different diseases. So exon inclusion the idea was from the nusinersen study. It's not exactly the same, but the biology is kind of similar. Like the idea behind it is similar. AAV9 is being used in gene therapy for, I think, ALS, yes? If I...
mentioned too, at the NIH, we have phase 1 trials for giant axonal neuropathy. It is AAV9 gene therapy.
Yeah.
And the gene therapy, the vector was created initially in academia, so University of North Carolina, Chapel Hill, then purchased by Bamboo Therapeutics, and then Bamboo is a small kind of biopharma. Bamboo was acquired by Pfizer, so there is interest by the bigger companies pushing forward gene therapy for rare diseases, and giant axonal neuropathy is actually much more rare than myasthenic deficiency. So there's precedence, and the gene therapy for the MTML myopathy, another muscle condition going forward if we launch in the
Right.
coming year. So a lot happening.
Yeah, exactly. And with the CRISPR itself, I think because it's such a, how do you call it? It's a hot thing. So everybody is like working on it.
Yeah.
And from our side, we will learn from all of these people. So right now it's a trial in LCA. Do you know what it stands for? It's an eye disease, Labor something CA. So from that, then I'm sure we will learn. We will learn a lot from this.
Do you see there being a chance that the brakes will get put on CRISPR at some point because of the ethics behind it? Like while we all have kids that we can see a great benefit from CRISPR, there's somebody else who wants to have a kid with blue eyes because of it, or they want to change something like make them look better. I see there's issues with that opens up a huge can of worms, of which I don't care about any of those worms right now. I care about my kid. But do you see there being a problem with CRISPR and the excitement building too much that someone's going to say, put the brakes on this, is this going too fast?
Personally, I think if we use it postnatally, so postnatal, not in embryo, I don't see a problem with it. Like maybe I'm too ballsy here, but as long as you're not changing anything in the embryo, I don't see why it's wrong.
I think for any of our therapies, they could be abused theoretically. So anything that enhances muscle performance or enhances muscle regeneration could be abused by athletes and so forth. So yeah, I mean, there are definitely ethical issues, and people are talking about that quite a bit at the moment. But I think, for the rare diseases and other diseases, we see, a potential path forward. There are issues with it, and the technology is still young, so we make sure that, the first thing, we don't want to do anything worse, right? we don't want to make anything worse, so we want to make sure that it's really safe and it's going to do no harm at all, and we hope then we'll get the benefits from it. And that's, I think, for all of us. That's why I think it's important to these technologies be duplicated in other labs too before we move forward and actually start even entering into trials.
I think that's a very key point of cross-validation in different labs of the same therapy or intervention that we are trying. And I think, there's always this big leap of faith from the mouse to human, right? But if we have at least validated so that we know that independently researchers have come to same results, I think that will give a lot more confidence for the clinicians to believe in our data and move it forward. And sometimes, again, it's money, 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 like, you know, to revalidate that.
Even the mice have variations. So
Right.
Sweta's mice actually died at a much younger age
Right.
than our mice, and it's simply because of the care, maybe, or the food we give them. And we actually just published a study showing the variations between 4 different
Right.
labs, international study, and it's quite a bit. The mouse, you know, your colony is different to some
Right.
colony of mice, and this the way you treat them, or what vitamins are in the food. For example, vitamin E.
Vitamin E.
Vitamin E was different, so our mice live a little bit longer. We have higher vitamin E in our chow. You know,
Are you based in New Zealand?
yeah. I mean, no, I'm actually in Reno. I'm in Reno,
Right.
Nevada, School of Medicine. Yeah. So yeah. I mean, these little variations sometimes bring up variation, and we didn't realize that the patient population has also got variation.
Right.
Right. So you know, it's
So,
one of those things you have to.
so that's also important, I think, and not only the same strain that has variation, we have 3 good, I mean, not many diseases have that. We have actually very good animal models to be able to cross-validate in these different animal models with different severity would also make a huge difference.
Yeah, I think it's really lucky. The mouse models replicate the human disease very,
Right.
very
Which
well.
is very rare, actually.
Yeah.
So it's a very, very fortunate thing. I just was being told by the
Yeah.
deep folks that are wearing lovely orange
Right.
sweaters that it's time for lunch. So I don't know if we could possibly, have any more questions?
I had just a couple quick questions. When you say gene therapy, does that mean replacement gene therapy? You're replacing the gene? Or are you tricking the body into thinking that the gene is still there? How does that work?
The second one.
That's the
So
second.
okay.
In her
Because
case.
In my case, yes.
You're tricking the body because
Yeah.
I know that in a former conference in Milwaukee, they talked about the different sequencing of these different diseases like Duchenne because this position at number 55 is missing.
Right.
It causes 58 and 59
Yep.
to cause the disease.
Yep.
Yeah.
So if you're able to trick the body into thinking that when present, it'll act normally.
Right.
What about LAMA2? What is the thinking there? Is it more likely to trick the body into thinking that the protein exists, or is there a way of actually delivering a gene?
Yeah. So.
Well, it's problematic because not everyone has the same mutations, right? So if everyone had the same mutation, and we've got a very kind of an ultra rare disease, then yeah, that would work because we could actually do a really informative clinical
Right.
trial. But because all the patients and all the children have different mutations, then it becomes a little bit more tougher proposition for exon skipping.
Right.
Try and skip over it and then re-establish the frame of the thing. For gene therapy, you're actually delivering a functional, normal copy of the gene with a virus, and it's
See,
Mm-hmm.
one thing that's kind of complicated with Sweta knows that very well is you can't package the entire gene.
Right.
and
Yeah.
it's difficult.
There's a really good potential solution.
Right. So I mean, so that's one strategy which in their lab is doing. You know, tricking the body to make the protein again by maneuvering the gene. But the other thing is that what recently a paper has come out of Dr. Marcus Rueg's lab and Peter Yoshenko's lab, so that what they are doing, they are putting another protein which can take the job of this missing protein, and that is gene therapy, just packaging that little linker protein and establishing the link between the missing, you know, so laminin is not there, it's not linking to important other proteins, so they are just putting that another surrogate protein and which can establish the link back, and that would be very, if it can go forward, it has lots of advantage because the body is already seeing that protein at some level, so that immunogenicity will be little less. So the results are extremely convincing, so it's a possibility.
One other quick question, but first just confirmation of an assumption, and that is it sounds like there's still a fair amount of underreporting in the population for myrosin deficiency. Is that accurate, or do we think well, if that is accurate, do we have a sense of how much it's being underreported?
Maybe
and
Pat.
what can we do as a community to increase the proportion?
Maybe Pat, do you want?
Yeah, I would say certainly. I mean, it's one of those things that falls under you don't know what you don't know. But I think everybody believes it's larger than certainly than what we have kind of numbers-wise within the registry. So I mean, that's numbers of 2,400. You've seen, different numbers come back.
What do you use? I use 150,000.
So that's just one. The second
Am I wrong?
is what they haven't
There's,
a lot.
looks like worldwide maybe,
Put these kind of things on as
well, in the
a
US,
fun
there may be 350 patients in the US. I remember.
There are 17 in
Ultra, that's why
Canada.
ultra rare.
There are 17 in Canada. That's what
There's only like a few in Australia. So,
But it's all of us that makes this happen, guys.
17 in Canada. 17
17
in the registry.
registry, yeah, 90.
This is impossible. So, and with other institutions like the NIH and others. So I think Pat's done a great job with CRISPR and the team. I mean, I can tell you, I know these guys. They work on shoestring budgets. I mean, they're trying to put 2 tin cans to make the Empire State Building, right? So, my hat's off to the research.
I'm,
I was going to say, all 3 of these folks up here have been funded directly from donations that came in through CMD. So and they do. I mean, they make it work on a lot.
I want to say, I think I would have long gone if not for Cure CMD. So my thanks to them.
I also wanted to point out
Thank
that
you.
I teach medical students, and I actually spent a week on the dystrophies, and actually their last exam is on Myristoyl deficient congenital CMD. The idea is to make it more so that they identify children at a younger age, so they don't get misidentified, because I know that's a problem. So I try to introduce them, and they get really excited. That's probably one of the top classes that they score on it. They love that. So yeah, it's a good course.
Thank you.
Thank you, everyone.